So let's talk about lipids now. So lipids are hydrophobic, they're nonpolar, and they usually consist of long hydrocarbon chains. They're also used covalent bonds. So let's talk about their functions. So one of their functions is insulation, another is energy storage, same as carbohydrates. They also have a structural function along with endocrine signaling. And then we can go on to the types. So there's a bunch of different types of lipids. Let's start with triglycerides. So triglycerides consist of 3 fatty acid chains along with 1 glycerol backbone. And they have 2 types inside of them. They have saturated and unsaturated. So when I think of saturated, I usually think of very highly concentrated or a lot of the substance. So this reminds me of how saturated fats are pretty unhealthy and they actually form the fat plaques. So they stack up on one another very densely, which shows that it's very highly concentrated or there's a high amount of it. And they have straight chains, so they can pile up on top of each other and no double bonds because of this again. Unsaturated, on the other hand, are not unhealthy, but because they have double bonds and they have bends. So when they try to stack on top of each other, they can't go perfectly in line like saturated, which would just go almost like this, right? Unsaturated instead will have the little bends, right? So they can't stack like that, right? I don't know how they stack, but they will not stack as densely or as packed as saturated. There will always be little spaces in between the sections. And yeah. Then we have phospholipids, which consist of 2 fatty acid chains, which are the non-polar side, and then the phosphate group, which is the polar side, and it is attached to a glycerol backbone. This allows this type of lipid to be amphiphatic, which basically means that it has a polar end and a nonpolar end. In that case, the nonpolar is the 2 fatty acids, and the polar is the phosphate head. So this allows basically the membrane to form spontaneously in water, because what happens is all the polar phosphate heads will attract to the water, and then the nonpolar will go inside. So it'll almost form like a little sphere with the nonpolar fatty acid tails on the inside with the phosphate heads on the outside, if you get what I mean. And this allows like that phospholipid membrane to be formed. And then we also have steroids, which is pretty interesting because they have 3 6-membered rings and then 1 5-membered ring. So there's 3 of them, which would be 6-membered, like so. And then there's only 1, which is 5-membered. So 3 hexagons, 1 pentagon. Does that make sense? And they're used for hormones and cholesterol and also vitamin D. Also, they're also for sex hormones and corticosteroids. Okay? So let's talk about lipid derivatives. Both phospholipids and steroids are technically lipid derivatives, but there also are some others. So for example, there are waxes. So waxes are fatty acid esters with monohydroxyl alcohols. And they're honestly used just how waxes are for protective coating on skin and exoskeleton, not human skin, though. And then we have carotenoids. I'm not that great at pronouncing it, but carotenoids, which are fatty acid carbon chains with conjugated double bonds. So they consist of keratins, xanthophylls, and they also have 5 to 6-membered rings at each end. So they're highly used for pigments in color in plants and both animals. So if you see a plant potentially yellowing, you know that they might be lacking some of these carotenoids, which I still cannot pronounce. And then we also have one other lipid derivative, which is called porphyrin, which are 4 joined chiral rings with a metal atom at the center. So think of hemoglobin, right? So like in the center of hemoglobin, there is an iron atom, or at the center of chlorophyll, there's a magnesium atom. I think I also missed one, which is the adipocytes. Just fat cells, technically, and they have two categories, brown fat cells and white fat cells. So the brown fat cells have a high amount of cytoplasm, liquid dry liquids, and tons of mitochondria. But the white fat cells, on the other hand, are triglycerides, so what we talked about up here, with a thin layer of cytoplasm surrounding them. Yeah. We also have glycolipids and lipoproteins, which are less common, but also very necessary to know. So glycolipids are phospholipids, except the carbohydrate group, there's a carbohydrate group instead of the phosphate group. No, wait, carbohydrate instead of the phosphate group. And then lipoproteins, which are lipid cores surrounded by phosphates and adipolipoproteins. And they're insoluble lipids. Help with transport, though. There's always an ideal state for the cell membrane and fluidity. So when it gets too cold or it's too rigid, then there's cholesterol and unsaturated fatty acids which are added to make the membrane more fluid, because unsaturated fatty acids pack less densely. Versus on the other hand, when it's too warm, then the membrane becomes too fluid. So you want it to be more rigid, so you'd add cholesterol and saturated fatty acids instead to restrict and stiffen the membrane. So that's lipids, and we continue on with proteins.